Hello everyone and welcome back to my YouTube channel. So today we are going to start a new topic, or you can say the new chapter of class 12 biology from the portion of genetics, that is chapter number 5, principle of inheritance and variation. So today in this lecture I'm going to be covering Mendel's experiment, his methodologies, and of course monohybrid and dihybrid clause. And if I get time, I will also finish the exceptions to Mendel's law. We have 3 laws. The exceptions to these laws are also important, so I'll try to cover all these things in this particular video. So this video is going to be super awesome. I just want all of you to pay attention, okay? So first of all, in this chapter, we are going to study about inheritance. Now, inheritance lana khalai. Inheritance lana, the process by which a particular trait sobo ki khejuji also ki pass on chigiyena from generation to generation. Okay, so suppose skin color roji chasha waina digiyode yamna earlobe kashi free yode kashi ki attached yode tibuji ga iris ki color migi chöni zo and the hair color. These are all genetic traits which are transferred from one generation to another through gametogenesis and after fertilization and zygote la pass. Okay, so the first topic of today's lecture, Mendel's experiment. The Mendel today, in the early time, okay, in the 18th century, okay, in the 18th century. 19th century, there was a scientist, there was you can say geneticist called Gregor Johann Mendel. Okay, so actually he was a priest, but along with that he was also a scientist. Okay, now till 19th century. Okay, so no one knows about the traits, trait flow chagiole. Okay, so that process is called inheritance, and inheritance was given by Gregor Mendel. Gregor Mendel ki kya inheritance did. Now, he accidentally did an experiment on Pisum sativum, or you can say pea plant. Okay, Pisum sativum manala, Pisum sativum manala chipal. Now, pea plant la chidu koi khaine resina in the pea plants. In the pea plants, the traits were passed in a particular fashion. Fashion chinala passed on chigidu. Okay, this chidu ni ko gi thangshu khare kindere monohybrid cross kini le. Okay, now before monohybrid cross, ko gi cross chigidu. Cross lana khare. Hybridization means mating. Mating means fertilization or syngamy. So, syngamy has to do sexual reproduction in the process of pollination, pollen pistil interaction. So, then methodology is successful. Now, methodology is method. Now, then methodology is okay, so methodology. The first method that is your A1 method or step is emasculation. You can emasculation. Okay, emasculation. Suppose emasculation is the process. Emasculation is the process by which the anther of a bisexual flower is removed. Suppose this is a flower and dinala. Okay, so this is the female part, that is your pistil, also called carpel. The carpel in which I show. Now, there are stamen yoda. Okay. Bilobe stamen. Okay. Now bilobe stamen yoda. Now this is a bisexual flower. It's a bisexual flower. Bisexual flower is also known as monoecious. Monoecious flower. Now monoecious flower nana casing also the part. This is your filament, this is your anther. If we remove the anther, or you can say the male sexual part of the bisexual flower, then that process is called emasculation. The emasculation is suppose diniga thupsare, okay, diniga male cheta dichipul thare. Now we have converted bisexual flower into unisexual flower. The step number the B1. B1 is your bagging. Now bagging is defined as the process of covering the stigma of the emasculated flower through butter, paper or plastic. In order to prevent pollination by unwanted pollen grains. That is the experiment schedule. The experiment schedule that the emasculated chesa that ovary gana so where the anther the malo the case chilo ne anther testicle pollination the conjunction chasi wind pollination chasi and it's a pollen grains also the pollen grain make you chilo the pollen grain sheba conjunction the gana possible. So the guys do also cut you the gana to the covering chikashilo the butter paper covering yamada plastic covering in order to prevent it from pollinating by other anther, or you can say other pollen grains, foreign pollen grains imagine so pollen grains are and that process is called bagging. Last one is the transfer of pollen. Last one is the transfer of pollen. Okay, so transfer of pollen means the pollen grain that we have. Now, कौन के experiment चुदी नहीं, कदा उनको कि pea planting की होता है, pea planting की होता है. So the case pea planting ना, pea plants are usually bisexual. Okay, pea plants is a bisexual. So the pea plant रहता है, लो क्या के experiment चुदी. Suppose this is your pea flower of the pea plant, that is your Pisum sativum. Now they like do, ता उनको कि emasculation चीज़ सारे, जरा bagging है चीज़ सारे, ता उनको कि खाला होता, उनको कि pea plant sheba so where the pea plant didela pollen grain di luda so also khat share suppose there pea plant sheba ji flower sheba dela anta yoda digala kya na and dela filament yoda dela anta na pollen grain yoda the pollen grain collect chi and digala to share ji covering to and dela also transfer pollen grain sheba this seed sheba digi male gani the definition ke jama uthi gidalo so just revisit the definitions clearly by rewinding the video. Okay. The transfer of pollen. Just to the methodologies which were opted by Mendel in his experiment. Because the point sumadi reason for choosing. Reasons for choosing Pisum sativum, or you can say Pisum sativum. Now, also if pea plant kachi ni choose chile, big reason is it here. Okay, reasons for choosing the pea plant. So pea plant choose chile reason khale la. So reason she originally kya reason ala she ma the chu mare. First one the it is bisexual, so you can say it is monoecious. And controlled cross. So the cross the controlled controlled cross is possible. Controlled cross is possible. Okay, controlled to gas is possible. Nivadi Kasana, it produces a lot of seeds. It produces a lot of seeds after fertilization. It is also called syngamy. Okay, syngamy, lot of seeds. This is the key word. Okay. Now it is an annual plant. This point is very important. It is an annual plant. Annual plant. Annual plant with a short lifespan. Annual plant with a short lifespan. Okay, annual plant it has a short lifespan. Now, short lifespan the result. Okay, experiments will be conducted and results will be shown quickly. Now, fourth and the very important point, it shows 7 pairs of contrasting traits. And the contrasting traits now okay, contrasting. A pair of contrasting traits is known as allele. Okay. So, is 7 alleles in the sense allele doing it pair doing it the trait mitawa doing okay, pairs trait the pair doing tolashina character doing okay. So 7 pairs of contrasting traits. Contrasting trait okay. Now so contrasting trait so these are the 4 reasons for choosing by Mendel. And then experiment first experiment is your monohybrid cross, which I will write here. Okay. Now monohybrid cross the dihybrid cross the Mendel experiment he did the experiment for 7 years from 1856 to 1863. This year is very important. Okay, from need point of view, both point of view but from need it's quite important. Okay, now monohybrid cross. Now monohybrid cross means P plant now P plant we are taking 2 P plants which are identical but differ in one character. Character differs also give monohybrid cross like that. Character differ okay. The example tall short tallness dwarfness. Best example is tallness dwarfness. Color but mostly also tallness color. So I will write here what he did was he took okay, so flower ni okay, flower ni emasculated chick male chick female okay, so flower flower female flower okay, bisexual bisexual unisexual so female flower P plant suppose female tall enough stem height female plant tall enough. Now tall in making kachi yore kogi allele nga khade pure le. Okay, pure homozygous tall len. Pure homozygous tall female flower which is breeded or you can say crossed with pure homozygous drop male ne dila female drop ne dile chick tall ne chick drop ne sha. Yeah, I say drop. Okay. Now here I just want to give some extra information here. That there are a number of gametes के लिए चुना. Is an only number of. Okay, number of gametes. कैसे सुविधा देना? Gametes की formula दिखा रहे 2 power n. Okay, so this is the formula for number of gametes. Keep that in your mind. Okay, number of gametes की formula दिखा रहे 2 raised to power n, where n is the number of heterozygous character. Okay, so heterozygous character kacho yuna digam se kya la gamete sugire. Now here how many heterozygous character are present? Nothing. Che da sama homo de. Homo ina n की value 0 है. So anything raised to power 0 is 1. So जो gamete ina sugire, that is T gamete. Okay, so pay the number of gametes actually it gives the number of type of gametes. So kya la tall there also gamete, and then kya la T chong gamete also. Okay, so these are your parents. Okay, parents. Now there are also gametes. You got the gametes. Now, the fertilized sin gamete. Now after syngamy, you will get heterozygous tall. Okay, this is your F1 hybrid. F1 hybrid. First generation Dihybrid. F1 hybrid. Now this was exactly what Mendel also got to see, or you can say he saw that in the first generation all the offsprings were tall. Okay, the genotype, this is your genotype. Genotype means the composition of gene is this one. Now phenotype, that is your expressed character khali physically expressed the khas is tall. Wow. So in the first generation 100% tall but pure tall hybrid tall. Now that these are the process selfing selfing. Now selfing is the process in which F1 hybrid F1 hybrid chick so mambo is some self fertilization china. Okay, self fertilization china okay, self fertilization chidu. That dirana kashi male kashi female the male ragyo dina female ragyo dina gamete because in this case you have one heterozygous character. Tallness the heterozygous character chick usually 2 to the power 1. 2 to the power 1 means the number of gametes 2 so male gamete means with capital T small t, female gametes with capital T small t. Now when we do the cross, there are colors ET, there are colors T, capital T small t, there are colors capital T small t, and there are colors small t. Okay, so there are phenotype laws. Now the box that we are using here in order to do such crossing was given by a British scientist, okay, called punnet. Okay, so this particular square is known as punnet square. Okay, so this particular square is called punnet square. Yeah, punnet square. Then punnet the scientific name. Punnet was a British scientist, English scientist, and British scientist, biologist, British geneticist or British scientist. Mark my words, this has been asked in your NEET exam. Punnet was a British scientist. Assertion reason is that. Courses were serving disability F2. F2 generation F2 generation and F2 generation phenotypic ratio okay, phenotypic ratio and genotypic ratio. Okay, phenotypic ratio and genotypic ratio. Now, phenotypic ratio express character density ratio. The tall, the dwarf. So phenotypic ratio F2 generation 3:1. 3:1. I can bracket na cut here. 3 can be tall is to dwarf ratio. Tall is to dwarf ratio 3:1. Genotypic ratio can 1:2, 2:1. Pure tall, that is your TT, 1:1. And here 2 heterozygous tall, 2:1. And last homozygous dwarf, 6:1. So this is what we call monohybrid cross. Okay, so so keep everything in mind because I'm telling you everything that has been asked in the exam. Okay, nothing more, nothing less. Okay, so okay, that is design ba. Okay, the class teaches this will show. Okay, so next one you can be Dihybrid class. Now parents differ in 2 characters. Character means okay, so parents differ in 2 characters. Dihybrid monohybrid differs the mono engine to tall the dog engine to color okay. 7 pairs of contrasting characters tallness and darkness is just one of the pair. Now in dihybrid class, the most prominent example is that this is your mother, that is your female, pure, tall, or you can say tall example but they should be differing in 2 characters. Suppose can be R, Y, which is Okay, so here in pea plant, when we talk about stem height, जैसे pea plant की case में हम जो stem height का नशे ये ना tallness is dominant, dwarfness is recessive. कुछ उसवा दी case ना ना तब character चिक दी stem height है. Character शंबा दिखाएं दिखा जैसे ना seed shape. मतलब नहीं हो रही seed shape दा seed shape is what character. And seed shape की form नहीं हो रही form नहीं देना खाने के लिए हम जो की trait लगे रहे. Okay, character देना ना trait नहीं दिखा दे. Round and wrinkled. The R, round the dominant J, that's why here capital R means homozygous round. So here you have to say R is a round seed. Okay, round seed. Or you can say round Lashana Dixere. And in case of seed color is the third character. And character and the trait is yellow seed and green seed. Yellow seed is dominant. This is yellow की capital written. So these are the round and yellow seed. What's this of round and yellow seed? The D case now green and okay. So क्या मतलब है? Round and yellow seed, green and wrinkled लगा है. Wrinkled नहीं है तो क्या मतलब है चिपक? Wrinkled seed. Okay, now they are homozygous. Homozygous means कहाँ ना थंगी formula चिट्टे सा formula ना n की value दिखाई चासो 0. So 2 की power 0 खाले 1. So obviously हम से घर से मिले 1 gamete सुशेगे और one type of gamete only, that is RY. And then जो यहाँ घर सुशेगे small ry. Okay, so these are your gametes. Gamete sushigde. The gamete sushadu. Usually fertilization chegde, syngamy takes place, and you will get small r, y, small r. Okay, so this is your F1 hybrid. F1 hybrid भी दी जाती है. Now F1 hybrid is also round and yellow. Okay, F1 hybrid is also round and yellow. Now next case is selfing. Selfing we need to draw the punnet square. In this case, the punnet square is a little bit more complicated, and I will also do today. Okay, so this is your punnet square. Okay, punnet square there. Now punnet square now na. Then now ki heterozygous character kazu. We have two heterozygous character. So heterozygous character ni hoy 2 raised to power 2. Kazu is 4. So female chigle, male chigle. Female digi she chigle, male ki gamete she chigle. So gamete she 100 chigle na 100. R capital R, capital Y, capital R, small y. Combination she has and then you have R, capital Y, small r, small y. So what's it now? Capital R, capital Y, capital R, small y, and then small r, capital Y, and then small r, capital Y. Okay, last one. Now cross theoretical. So R, Y, Y. So what's it? Capital R, Y, small y. So what's it now? R, small r, Y, capital Y, R, small r. So capital R, small r, capital Y, Y. So again, similarly, you have to fill these things. small r y y, r y y, and then you have small r y y, and then we have r y y, and then we have r y y, and then you have r y y. तदी कांच है बा. Now, कोशिश अच्छे दाने main formula दिखा शीश है. Now we have filled the punnet square. Now it's time to write phenotypic ratio and genotypic ratio. Okay. So in this case, that is your dihybrid cross. Dihybrid cross case na. Okay, in your dihybrid cross na, here the phenotypic ratio. Okay, ratio of the express character दिखाजु चाहिए उसे ना. 9:3:3:1. ठीक है यहाँ पर. Now 9 ना this 9 din 9 दिखाई फिर क्यों रे? din 9 दिखाई फिर क्यों रे? R Y Y. सामा capital U की condition देंगे. Homozygous dominant. This okay, this one की condition दिखाई फिर क्यों रे? Homozygous green and R Y. Now this one की खरे new combination. Okay, parent this one किधर नहीं बने. Parent चिपक चिपक ठहर रखे हैं ना offspring थी. वो you can say progeny. ये ना है. These are your F2. Okay. F2 generation. Now 3:3 na, the first 3 symbolizes R okay. So R Y Y then. R Y Y this way so tipo chique de unser parent this way. Okay. Now this symbol has such yours na. They be round and green. Chique round and green symbolize here. Okay. Just somewhat na. The first three cutting your round and green. New combination. Parent na round and green do window round and yellow round and green sawa. And 3 shema dekha cutting your wrinkled and yellow. And also genotype na room mare. So kya ki R Y Y the pure de that. The pure thing here. Pure round and yellow. And then the pure the genotype on the sheet and any mother than you. 9 they can help you round and yellow. Okay. 1 they help you wrinkled and green. So this is good. So 3:3 the new combination. In the can you also large and 3 or the okay, so phenotypic ratio. Now phenotypic ratio is jela also the kali is genotypic ratio. Now genotypic ratio is very much complicated and it is not asked in exam. ये क्या है ठीक है होता है. ये जो मुहाब होना चाहिए इसे ना रहने में देता 1:4 2 1 2 1. So 1:1:4 2:1:1 is your genotypic ratio. It is quite complicated. It's a need okay in our tissue. Not that much important, but this is quite important. Phenotypic deification. Okay. Okay, so after his monohybrid and dihybrid laws, Mendel gave three laws, okay, which are collectively called Mendel's laws. Laws of genetics and DNA. Okay, so first law of Mendel, law of dominance. Law of dominance. And the second one, law of segregation. Segregation. 那同时我们就可以出 definition 的时候，so law of dominance 你看到一个的 it states that only one allele, or you can say trait, allele की trait control चाहिए, ये जो तो ये बात हम बता रहे हैं, okay? Allele controls trait and gene controls character, okay? तदा आज लाइट में जिसको Mendel का जो तदा आज की फिर character जो रूम में pass चीज़ और inheritance सीखेंगे, तो gene lag की term delay हो मारे, so Mendel की दिला खाई मिंग जितना शे, तो तदा आज gene lag दिया जिला Mendel की मिंग खाई तारे factor ताशे. Okay, so gene is also called Mendelian factor because Mendel gave main factor. Okay, so move on to the next one. Dominance. Dominance it shows that only one allele or trait of the parent is expressed in the F1 progeny in the F1 progeny in a monohybrid class. Thus, the unexpressed trait is called recessive. Okay, so Mendel's first law of dominance. Mendel's law of dominance is okay, so law of dominance equation is only one allele or trait, allele or trait of the parent is expressed in the F1 progeny in a monohybrid clause. Did I think of the dose and but monohybrid clause nada to express phenotypically expressed and the expressed one is called dominant and the unexpressed trait is called recessive. Okay, unexpressed trait la recessive. So the example you have to example you have to can be tallness and roughness functionality. Okay. The second law, that is law of segregation, it states that allele segregate means separate during gametogenesis. During gametogenesis, that is your formation of gametes, sperm down over soil, pollen down over the egg soil. Gametogenesis and combined during fertilization. Okay, alleles segregate during the gametogenesis. Chick-chicken hey, the gametes to the allele must segregate chi mother. Doing what gametes to do. And general langya combine chamase. So they are the law of segregation. It states that alleles segregate during gametogenesis and combine during fertilization. Or you can say syngamy. Okay, hence there is no blending. Hence there is no blending of character. Hence there is no blending of character. Hence there is no blending of. Hence there is no blending of trait. Okay, hence there is no blending of trait. So these are the three first two laws of Mendel. Mendel the law of Tangshini. Okay, so law is only monohybrid cross. Okay, the first and second law are the result of monohybrid cross. Okay, monohybrid cross is result first and second law. But the third law is the result of dihybrid cross. Okay, the third law is a result of dihybrid cross. And the third law is law of independent assortment. Independent assortment. Okay, the third law is law of independent assortment. The law of independent assortment, the dihybrid clause need to do. It states that different pairs of did this to confusing it. Too much did a segregation like this to confuse everyone. So it states that different pairs of allele, okay, it states that different pairs of allele segregate independently okay, it says that different pairs of allele segregate independently and then assort independently through syngamy. Okay, so the law of independent assortment the allele pair into monohybrid segregation the allele pair now independent assortment allele pair ni delay ma okay, when two or more pairs of alleles are present together, the action together. Okay, different pairs of alleles segregate independently and then assort independently throws in gametes. So, if the page now, then also give two different, then also the pair has the pair need pair allele pair need that is round pair and yellow pair, or you can say seed color ki allele pair and seed shape pair. Ini pair pair ni yodu and kya ki dela yapo chintana monohybrid dog chalo de R the twinso. Chikwachikyam R the twinso. So color ki R the Y the kinki R twinkle the lano ki yomala chikwachikyam R the kinki Y lano ki yomala. Tanda color sena Y R the mena ni only monohybrid dog chalo de Y takka twingido. Chikwachikyam Y the medio R chikwachikyam twingido. So color ki sena allele ki pair chigi allele shamba ki pair dilo. One pair of allele will not interfere with the segregation of other allele. Okay, allele shamba ki segregation nala interfere chigi mari. This is also the independent assortment lie. Okay, otherwise the definitions are written here. You can note it down. So I think that's the end of this lecture. In the upcoming videos, I will cover the exceptions to Mendel's law. But right now, I just want to tell you that law of segregation has got no, no exception. That's why law of segregation is also known as law of purity. Otherwise, we do have exceptions to law of dominance and law of independent assortment, which we will cover in the upcoming videos. Okay? So till then, keep studying hard and don't forget to like, share and subscribe. See you soon in the next video.